Hi everyone. Welcome back to the third part of this lecture, which is about a method for estimating heritability using individual level data. So I'll be talking about two very popular methods, the Haseman-Elston or HE regression method, and another method, which is the genome-based restricted maximum likelihood or GREML method. So those two methods are implemented in the software GCTA, which we'll play with during the practical. So let's start with the Heisman Elsin or HE regression method. And so this is a regression method in a sense that it directly tries to estimate heritability by regressing some quantity on another quantity. So the first quantity, which is called Zj, Zk here, that quantity is exactly the product for all pairs of individuals in the sample. So for individual J and individual K, for all those pairs, we calculate the ZJs as follows. So that is that we take the phenotype and we essentially normalize it by subtracting the sample mean, the mean in the sample, and the standard deviation in the sample. And the reason why we do that is because the expectation of that sort of standardized phenotype is exactly the phenotypic correlation. So that starts to ring a bell. And so what we're doing here is that we're regressing this product, cross product or product for all possible pairs in the sample. We are regressing that product onto the pi hat JKs, which are the entries of the GRMs. So let us now just illustrate that with using some UK biobank data that I already shown you in the previous part. So what you're looking at here is on the x-axis, the pi hats for a set of relatives. So the third degree, second degree, pi hat around 0.5, remember, is a combination of parent offspring and also full SIB individuals here. And on the around 1, we have exactly the monozygotic twins or MZ twins. And so the ZJZKs on the y-axis are calculated using height by subtracting the mean, the standard deviation. I actually here also subtracted the differences between males and females, just to make things a bit more neat. And so what HE regression does is that essentially perform this ordinary least square estimation or regression and comes up with two estimator. So we have an intercept and a slope. And so I'm not going to comment too much on the intercept in this part of the lecture. I will touch on that a bit later in the next part. But here, if we just focus on the slope here is exactly, it gives us an estimator of heritability. And in this case, it gives us an estimated heritability of 0.87. So the HE regression, so the example actually that I just shown you is it's everything that I performed in R. Actually, I had about 98,000 pairs of individuals, which is manageable, and I could load everything in R. I'm sorry, I could load everything in R and run this regression using the LM function, for example. But in practice, it's not always manageable to do that in R. So you like to use softwares like GCTA. So, and how to do that with GCTA, actually, it follows about 3 steps. So first step is to calculate the GRM, right? So GCTA, fortunately, can read data in Plink format. So the Plink format you've probably heard about in the GWAS lecture. And so you can directly use B5, which is the same command as in Plink, sorry, B5, my Plink format data. And then you have this make GRM bin flag, and this flag essentially saying the bin because you want the GRM to be stored in a binary format. There are alternatives. You can use GZ for gzipped, or I think you just you can see more in the documentation of the website, on the website. But this one here just saying create a GRM and store it in the binary format, and out is giving the prefix of the output file. So in this case, GCTA will generate 3 files. A .grm.id file, which contains the individuals that you have in your input data file. You have this mydata.grm.bin, which will contain the actual values of those GRMs. So this is a fairly big file. And the other one is my data .grm.nbin, which contains for all pairs of individuals the actual number of SNPs that has contributed to calculate the GRM. And so that number can vary between people because of missingness in the data, right? So some pairs of individuals may have more SNPs actually genotypes and et cetera, that can vary. So that's the step one, which essentially can just line one comment. And the nice thing is that once you calculated your GRM, you can store it and you can reuse it for a bunch of analysis, a large number of analysis you want to do subsequently. So the step number two, well, it could be the first one actually, is to prepare your phenotype. And what GCTA needs is a file with just like Plink, two first an ID for the family or an ID for the individual. So in this case here, I've used the same ID multiple times for the individuals, but what GCTA does is that if you uniquely identify the person, it actually looks at the combination of family ID and individual ID. So it doesn't matter here in this example that I have ID one the same value here or here. It doesn't matter as long as the combination of family ID and individual ID is unique. And so then here I just created two phenotypes, just now put in some number, and also the headers are not necessary for GCTA because you can specify which phenotype, the order of the phenotype you want. And so the step three, the third step, is simple. It's just now once you have your GRM in your phenotype file, you can just run GCTA -GRM, and you specify the prefix for your GRM that you just created here. --pheno, you specify the phenotype file that you've created. --mpheno1, which tells GCTA that you want to analyze the first phenotype. It could be two if you want to analyze a second, etc., etc. And then you add this HE-reg flag, which tells GCTA that you want to run an HE regression analysis. And so because this is a regression analysis, is a super fast analysis as compared to the other estimator I will talk about in the next slides. But essentially, when you run that, it runs very quickly and generate two files. So you have the log file, or when you can trace back some of the calculation, that GCTA does in terms of making sure that you've read the right number of individuals, and it's always a good thing. And this is a general comment. It's always good to have a quick look. Always have a look at the log file, and it generates the HE-reg file, and here just an extract of that file. So in which you have the intercept term and the heritability term. So it directly writes it as the VG times VE, which is essentially the variance of the genetic value divided by the VP, the phenotypic value. So essentially the heritability. And in this case, it estimates it about 0.4. So this is simulated data. And so HE regression, the way it is implemented in GCTA, actually implements two types of estimator. One is the cross product, the CP, which is exactly the one that I told you about, right? We normalize the phenotype, and then we create for all the pairs the ZJs times ZK. So this is the normalized phenotype, the cross product. But HE regression in GCTA also implements the squared deviation, which is regressing the ZJ minus ZK square on the GRM. And we can show that it gives exactly the same estimate most of the time. And just that the intercept doesn't have the same interpretation in this case. So I will, I think as a general, you can contrast both, but I think you can probably just focus on this HECP in this case. Here we go. So now I'm going to talk about the second estimator, which is one of the most popular method for estimating heritability using SNP data. Well, actually, the GREML is just about it can be, it doesn't have to be SNPs, to be honest. It just, as long as you have a matrix of relatedness, or whether that matrix of relatedness is calculated from SNPs or from expectation based on IBD, it will work essentially the same. So I'd just like now to break down the GRML as a method into two parts. So there is the G part, which is the genetic part, and in which I will spend a bit more time to explain. And there is this REML part, which is the estimation part, which is more general. A restricted maximum likelihood, or a REML, is a class of estimators that exists outside genetics. So you can find it in the literature, in the various, in the statistics literature, in social science. It has nothing to do with genetics per se. So, but I like to separate those two things just so that we have a clear understanding of what is important to understand in terms of the assumption of the GRML model, and the other part, which is just the estimation part, which can vary depending on the more general considerations. So let's start with the G part in GRML. So remember, in the part one, I introduced heritability using a model where the phenotype can be decomposed as a sum of two terms. So the genetic factor G and the non-genetic or environmental factor E, right? Y equals G plus E. And heritability was defined as a ratio, right? The proportion in the phenotypic variance that comes from genetic factors, so sigma square variance of G divided by variance of Y. And if we assume that Y as G and E, the genetic factor and the environment, are independent, if they are independent, then the variance of Y can be essentially written as the sum of those two variants, right? So this is just a recap from part one. And also to just like to introduce a few notations is that now on I will refer to sigma squared G as the variance of that. So this is the variance of that genetic value across all individuals in the population. And that's this variance of that environmental value, sigma squared E, across all the individuals in the populations, So one of the comment that I made at the time, in part one, was to say that the definition, that definition wasn't very useful unless we can observe G. And so having SNPs is actually a great opportunity to observe what the G could be, what could be the contribution of genetic factors. And so the GRML estimates heritability by actually modeling G. And the way it does that is by assuming a linear function of SNPs, saying that the G is a linear function of all the SNPs, which is written in this equation. So what is Gj? Gj is the genetic value, or the combination, the resultant of all the genetic factors, or I would call that the genetic value of individual J. And what I'm saying here is that the genetic value can be written as a linear combination of all the SNPs. The M SNPs I have available in my sample, I essentially sum across all the SNPs. And what am I summing up? I'm summing essentially the minor allele counts that are normalized in a funny way, right? This is the minor allele counts, which I introduced before in the previous part when I talked about the GRM. And so this is takes value between 0, 1, and 2, minus 2 times the minor allele frequency, pi, and are normalized by the square root of 2p. So this normalization actually comes because it's handy, and because under Hardy-Weinberg equilibrium, it essentially leads to the variance of that term to be 1, which is practical for the algebra and some of the considerations. And so essentially you have the effect of the SNPs, the genotype, it's just essentially the normalized genotypes, times an effect. So what I'm saying is that the genotype, genetic value of individual J is a sum of genotypes times the effect of the genotypes. Right, fair enough. However, there is a challenge, right? Okay, when I say that, to be able to estimate the variance of GI for all individuals, I need to know what are those essentially SNP effects, right? I need to know what are the UIs. And the problem is that although it looks like a regression model, multivariate regression model, it is actually a very nasty regression model, because most of the time the number of SNPs M is often, if not always, much larger than the sample size. So this leads us to regression models that are called underdefined, meaning that we essentially don't have enough information to estimate the contribution of each SNP. Right. So that's the challenge. But this challenge, sorry, has a solution. So the solution is to say, okay, I cannot estimate the SNP effect per se, but maybe I can, if I assume the SNP effect to be random, maybe I can estimate a property of their distribution instead of estimating them directly. So let's just follow that reasoning and start to make some assumptions about the distribution of SNP effects. So we're assuming that SNP effects are in fact random variables, and we assume that they are independent. And we'll come back to that, some of those assumptions in our next part, because we're talking about, in the part 4, we're talking about the interpretation of our estimates, and the interpretation derives from some of the assumptions that we're making. So here we're assuming that effects are independent. We're assuming that the effects are normally distributed with a mean 0, essentially saying that sometimes the minor allele will increase the phenotype and sometimes the minor allele will decrease the phenotype, but on average it's 0, which is fair. And we're introducing essentially that the variance of those effects is essentially the genetic variance, sigma squared G, divided by the number of SNPs, right? Each SNP contributes the same in this case. So under these assumptions, what we can show, and so there's a bit of algebra, but it's not very complicated, honestly. What we can show is that the variance of G, so G, this now I'm using the bold notation. So G bold is a vector, as you can see here, is a vector of all the, stacked together all the genetic values in a sample. So this vector contains things that we cannot observe because we don't know what are the YGs, but we can still write the variance of that vector. So the variance of a vector is a matrix, right? So we have n individuals in the sample. So the variance of G is going to be an n by n matrix. And so that n by n matrix can be written as sigma squared G as a scalar, which multiplies another matrix. And that matrix, if you pay attention, we can recognize that matrix is exactly the GRM, right? For example, for individual J and individual K, we're saying that we are averaging the sum of those ZI, J, ZI, K, and the way it was defined is actually exactly our definition of the GRM. So we're saying that the variance of the genetic value can be written as sigma squared G times the GRM. So, okay, one step further, but we're not there yet. We need another assumption, and the other assumption is about the residual part, because to be able to estimate a heritability, we not only need to estimate sigma squared G, but we also need to estimate sigma squared E, which is the variance of the non-genetic effect or the environmental effect. And so what we're doing here is that we're going through exactly the same reasoning as before, and we're assuming that this for all individuals that their residual values are normally distributed with a mean 0 and a variance sigma squared E. And by combining all these assumptions together, we can write that the vector phenotype, so which is the sum of G plus E, right? So this vector of all phenotypes which we observe, right? So Y bold, sorry, I'm pointing with my finger, but you can see Y bold, this quantity here is a vector that things we can observe. And so what we're saying is that the distribution of that vector is a normal distribution, multivariate normal distribution with a mean 0. Okay, which is fair. It's not a big deal because we can always subtract the sample mean from those values, so that's not a big deal. And most importantly, we're saying that the variance covariance matrix of that vector is equal to sigma squared G, the genetic value, times the variance of the genetic values times the GRM plus sigma squared E times IN. And why is IN is essentially the identity matrix. So it's a matrix n by n matrix. With a diagonal equals to 1, and outside the diagonals you have 0, right? And so with that model, so this is essentially the G part in the GREML. It's to come to that equation that we're looking at. And with that model, if we can estimate sigma square G and sigma square E, we can subsequently derive an estimator of the heritability by essentially dividing this estimator sigma square G by the sum of those two estimators. So that concludes the G part in the GREML. And now we're going to talk about the REML part. And I'll try to do that a bit quickly because, as I said, this is not intrinsic to the GREML part, where I think we've done the heavy lifting already. And the REML part is just saying, okay, now that we have a model with a distribution, how can we estimate the parameters? Well, we can use various a set of techniques, and restricted maximum likelihood is one of them. So let's start with the maximum likelihood part. So the maximum likelihood part is a maximum likelihood is a general principle in statistics to estimate parameters. And the idea behind this principle is to say that we have observed a set of data, and we assume that this data that we have observed was generated by a probability distribution. And that probability distribution is unknown, but we're assuming that this probability distribution lives in a family of other probability distributions that are indexed by a set of parameters. So if we change a parameter, you still live within the same family of probability distribution, but it's slightly different probability distribution. And so the principle of maximum likelihood is to try to explore all possible parameters, all possible index in that distribution, family of distribution, and find what is the combination of parameter that maximizes the fit between our observed data and our postulated probability distribution. And so this is example here, a very simple example where we have some data which we can represent the histogram from, and we imagine that we want to estimate the mean of that distribution. And so you're saying here that my family of probability distribution is a normal distribution with mean unknown, which I want to estimate, and a certain variance, which I assume in this case to be observed. So what we can do now is that we can vary on the x-axis all possible values of the mean. So again, we want to know what is the mean. So we just scrolling here in all possible values, and on the y-axis we have the likelihood corresponding to changing those values. And so what we see here is that as we change the value, the likelihood increases, reaches a maximum here, and then decreases. And the maximum here is not that far from the sample mean, which essentially is saying that the sample mean in this case is the maximum likelihood estimator. So apologies again, that was very quick, but I think that's just to give a refresh of some things you may have heard, and if you need more details, I'll point you to read a few more papers on that. And actually, Wikipedia does a great job at explaining those concepts already. So now the question is, okay, maximum likelihood we see, but why is it restricted? What restrictions are we talking about? So, well, before restricting, we need to augment first, right? And so the previous model I introduced was a model where the phenotype Y was expressed as two things, right? We have Y equals G plus E, genetic and environment and non-genetic factors. But in practice, we know that the data that we collect is not a random collection in the sample. We have ascertainment in the sample. We recruit people from specific centers near hospitals, for example, or within, I don't know, university campuses. And so we know that we have some experimental variables that kind of create some mismatch between our sample and the population. And again, what we want to estimate eventually is the heritability of the trait in the population. So when we know those experimental variables, what we can do is to account for those experimental variables in our analysis. And those experimental variables are also known as fixed effects. And here, this model that I'm showing now, here is a model that essentially expands the previous model by now including some of those experimental variables as covariates. And so we could have gone through the same idea of also assuming a distribution of those on those experimental variables, but in fact, in this case, we don't need to do that most of the time, because remember, the reason why we did that for the SNPs is because of the dimensionality issue, where we had a very large number of SNPs, and that was not manageable to estimate their effect individually. So in the case of those experimental variables, we can actually estimate their contribution specifically. And so in which case we essentially just keep them on this part of the normal distribution, on the fixed effect part here, and estimate them as a covariate, just as what we would have done in the classical, say, linear regression, for example. So this type of model, where we have some parameters that we estimate, the effect of some covariates that we estimate as fixed effect, and the effect of other covariates that we estimate a random effect, in particular, from which we only estimate the variance part, as we're doing here. This type of model is known as a linear mixed model, right? Linear because of the relationship between covariates and the outcome y, and mixed because we have this kind of two types of treatment, right? Differential treatment for fixed effect and random effect, and which comes about now that we have expanded the model. Why are we talking about this restricted maximum likelihood? Is essentially to say that the restricted maximum likelihood is the same as a maximum likelihood when there is no experimental variable, when there's no fixed effect. Those things are essentially the same. But when you have this fixed effect, the restricted maximum likelihood is a maximum likelihood strategy that is applied to a transformation of the data, right? It's not directly applied to the data, it's applied to a transformation of the data. And just to conclude on this, in practice, when the sample size is large, the restricted maximum likelihood and the maximum likelihood are highly concordant. So unless you have to estimate, I don't know, 100,000 fixed effects, if your sample size is of about 100,000 and you have 20 or even 100 or 500 fixed effects, restricted maximum likelihood and maximum likelihood will be fairly concordant. So the GREML, not regression, the GREML method is implemented in GCTA. And using a method, a command line that is very similar to the HE regression. So this is actually exactly the same. The only difference here is that instead of using --HE reg, I use --REML instead. And so what GCTA will do, it will generate two files, a log file, again, check the logs, and the HSQ file. And here this is an extract of that file. So you have multiple things. So you have the sigma square G, the variance of G, the variance of E, and their associated standard errors. You have the variance of P. So this is the variance of the phenotype. So it's the sum of those two things, VG plus VE, and the associated standard errors. And we also have the heritability estimator here, which is essentially this VG divided by VP, by the sum of those two things. So we have this one here, that's our heritability estimate. And so this example is exactly the same data that I used, simulated data that I used for the HE example earlier. And you can go back to that, but you see that the estimate is essentially the same. And so we have other things here. This is the likelihood, the maximum likelihood reached, or the restricted maximum likelihood reached. We have the likelihood if we assume that there is no genetics. So this is a constrained likelihood when we assume that sigma square G is 0. And the reason why we're doing that is because we can implement this likelihood ratio test, LRT, essentially to test the significance of the heritability. So this is what GCTA does, is it output variance components, variance of G, variance of E, the heritability, but it also tells you whether your estimated heritability is statistically significant or statistically different from 0. And this is the p-value that you can use for that test. And finally, you have the sample size. Okay, so just to summarize, so this part was a bit longer than the other parts, but it's quite central because it's you will need to know those concepts and those different methods for the practical and hopefully for the analysis that you'll be running on your side and just in your research in general. So in this part, part 3, we have introduced 2 ways to estimate heritability in a sample of individuals, knowing 2 things: knowing their phenotypes and knowing their GRM. Right? And so those 2 methods are HE regression, which regresses the sort of cross product values or normalized product of normalized phenotypes onto the genetic relationships, and the REML, which uses trying to model the genetic value of individuals by assuming that genetic value to be a linear combination of all the SNPs we have, which essentially amounts to modeling the covariance between individuals using the GRM again, and then apply a maximum likelihood to that derived model. So in the next part, we will discuss important aspects about the interpretation of heritability estimated from SNPs. And we're also going to talk about some of the biases affected this method and which will help us improve our interpretation. So thanks again for your attention. I know this part was a bit longer, but take your time, go again if needed, and we will be interacting anyway during the workshop, and I hope to see you for part 4. Thank you.